All right, so thank you everybody for coming here. My name is Giovanni Cavaletto. I'm with Index Fresh, and we have a pretty exciting agenda for you today. For those of you who don't know, Index we're an avocado packer shipper. We've been around for just over 100 years. Index is owned by a broad base of California family farmers, and with the goal of maximizing the value of the avocados that we handle for the growers that we work with. And one of the ways that we try to add value has been through doing these seminars. So we started about 4 years ago. The idea behind the seminars is to identify a topic that's important to avocado farming, invite 2 or 3 of those experts in that topic to come give a presentation to the growers that we work with, and then we do that presentation. We did it yesterday in Fallbrook, today here in Oxnard, and tomorrow we'll be up in San Luis Obispo. We record the presentations and place them both on our website as well as on DVDs that many of you have seen the copies floating around the county. So today we're really excited to be doing today's presentation in conjunction with the California Avocado Commission, kind of co-sponsoring this. We've got 4 speakers, so that's why I wanted to go ahead and get started. We do have a full agenda. The first 2 are going to be from the California Avocado Commission. We've got Ken Melban and Tim Spann to talk specifically about the current polyphagous shot hole borer outbreaks that are in California. And then we have invited 2 distinguished gentlemen from Israel, Professor Stanley Freeman and Professor Zvi Mendel. They are both with the Volcani Institute. Professor Freeman is a plant pathologist and Professor Mendel is an entomologist, and they will talk to us about similar shot hole borer outbreaks that they have been dealing with over in Israel for several years now. So with that, I'll pass it over to Ken.
Thank you, Dana and Giovanni, for inviting us to come and give an update on what we're doing at the California Avocado Commission, specifically to the polyphagous shot hole borer and Fusarium dieback. I think for many of you, you'll hear a lot of what we talk about this morning. We just had grower meetings 3 or 4 weeks back. Not a lot of new information, so we will go through it rather quickly, but we wanted to at least provide an update on where things stand now and what our current activities are. This is a good backdrop to start with. It demonstrates the funding that the Commission Board has committed to this pest complex since April of 2012 was when the first funding was approved. And if you run all the way through this, you've got projects that are multi-year projects, even including this most recent project that runs through 2018, for a total of $1.7 million. So your board of directors has not been hesitant at all to move quickly and fund where necessary research activities to attempt to find some sort of solution or at least remedy to mitigate the impact that this pest complex presents. I'm going to talk briefly now about the history, the movement, and our detection activities across the state. Most of you probably know this, but it was first detected in California in 2003 in the Whittier Narrows area of LA County. Unfortunately, it was misidentified. It was at that point thought to be Eualacia fornicatus, which is the T-shot hole borer. It was then subsequently captured a few more times in LA County on those CDFA yellow sticky traps, but again, it was misidentified. In 2003, there were a significant number of box elder trees in Long Beach in an urban setting that were infested, and there was tremendous damage caused there. But it wasn't until 2012 in a backyard avocado tree where it was properly identified in Southgate. As you all are aware, it's got a tremendously extensive host range. If you look here, there's over 300 trees that are known to be attacked by the beetle. Of those trees, 138 have been confirmed infested by the fungus. So it's got a huge host range. If you just look at the list, you know, box elder is up at the top, big leaf maple, sycamore, castor bean, and those are found extensively throughout Southern California. And it's not limited, obviously, from that list to agriculture. There's a tremendous threat, and it's already yielded a tremendous amount of impact in some of the urban settings. We talked about Long Beach a minute ago, but also just recently, University of California at Irvine discovered they've got a tremendous infestation there. This is a press release that was issued about a month ago, and I've highlighted a couple of areas because I think it's important that we in agriculture look at the urban setting where they don't have issues in terms of residue for food use on pesticides, et cetera. So they have a wider range of materials at their use, but they're not finding any materials that are effective. As I have highlighted here, it says there is no effective treatment for the severely infested trees. In the release, they go on to say that the shot hole borer may have been present on their campus for 2 to 3 years. As in previous cases, it was misidentified. It wasn't recognized as a shot hole borer. At present, they are anticipating having to remove 1,000 trees of their 25,000 trees on campus. Some of these trees are 40, 50 years old. It is going to significantly change the landscape of the campus. I think the only bit of maybe silver lining I would try to identify with this is that unlike the Asian citrus psyllid, which I know many of you are having to deal with on citrus, the shot hole borer has a wide reach across, you know, urban landscape also. So we're hopeful, and we're having conversations and meeting with other interested stakeholders that hopefully this will raise the attention on the threat that this invasive pest presents beyond just avocado growers. This is a map that shows the known distribution as of December 2012. You can see it was focused right here in LA County, and then these blue triangles are confirmed positives in 2012. Again, that was not in any avocado production areas. It was strictly in the urban landscape. A year later, you can see the boundary has now spread. This is December 2013. The red triangles are the positives, and you can see it's just worked its way out. Again, entirely in the urban setting. And then this was December of last year, and we now have the introduction in San Diego County. This is actually a second beetle. It's morphologically indistinguishable from the LA County beetle, but there are some differences in the disease that it vectors, and Tim Spann will be talking about that shortly. The other part of this is in August, as I'm sure you're all aware of, last summer was when the first detection occurred in an avocado grove in Escondido. To date, there are 14 groves that we've identified as infested, all within the San Diego County up to the border there of Riverside County. So your board quickly responded by funding the development and deployment of traps. We have to date developed a very extensive detection grid of these funnel traps here that you'll see on the right, and that is the lure cruciferol. To date, we have about 64 traps that are deployed in San Diego and Riverside counties. These are strictly for detection of new finds of the beetle. In your county here, we have 50 traps that are being monitored weekly. Thankfully, there have been no finds in any of those traps. In Santa Barbara, we have 5 that are under our control and 5 in San Luis Obispo. I want to point out that these traps in Ventura and San Luis Obispo and Santa Barbara are not primarily deployed in avocado groves because we recognize, based on the previous movement of this pest, that it will likely not first occur in an avocado grove if it's the LA County population. The chances are you're going to find it in a campground where, you know, campers from Orange County, Los Angeles, San Diego County, where there are known infestations, have come up to camp and they brought firewood with them that might be infested. We're also looking at nursery facilities and green waste facilities where we know there's a lot of importation of either green waste material or nursery stock that poses a great risk for hitchhikers. This is a map that was developed in collaboration with Dr. Keith Escalon at UC Irvine. The link here is on the bottom. It's an interactive map, and it shows, as we just talked about, the red dots show where the LA County infestation, LA beetle infestations are. The blue are the positive for the beetle and fungus in the San Diego County. And all of these yellow stars denote where we have traps placed as part of our detection grid. And I want to call your attention to this perimeter here, if you will, that we have intentionally set up. That is basically on the line of Los Angeles County and Ventura County. We really believe that an early warning will serve us and serve you best, so that we know that you can increase your vigilant scouting for the beetle if you know it's now crossed into the county. At the same time, we sanction pesticide and fungicide trials. We're working in 2 locations. One is at an infested grove in Escondido, where we've actually brought a field trailer onto the grove, and we've set up a lab there where the bioassays are conducted. We've also done some treatments at the Pine Tree Grove there off the 126 here in Ventura County. And you might wonder, well, how can you conduct any trials in a non-infested area? So what we're doing there is we're doing our infusion trials there. The trees are being injected with different materials that we're evaluating for efficacy. Then we're taking samples of the wood from those trees. We're taking them either to UC Riverside in a controlled quarantine lab, or we're taking them to our field lab. And then we are introducing the beetles to the material to determine if any of those pesticides and fungicides are efficacious. The 2 materials right now that we are gearing up to pursue a Section 18 for include tilt, which is propiconazole. We look at that as a possible Fusarium dieback preventative. It's a tree injection. The challenges there is it is very labor intensive. And then secondly, Hero. This may reduce beetle levels and levels of new attack when there is an existing infestation. In no way do we anticipate that it will fully cure an infestation. This is applied through a foliar application. The problem with that material is it will likely require up to 5 treatments in a season. So again, very, very costly proposition. At this point, I'm going to hand off to Dr. Tim Spann.
Okay, good morning. Thanks, Giovanni. So as Ken said, I'm going to cover a little more in depth the research that's been going on by the commission. So the research that we've been funding has primarily focused on identifying the beetle. As Ken mentioned, it was misidentified initially. So identifying it, figuring out where does it come from originally, has been a major part of the work that we've been funding. The basic biology of the beetle, understanding its life cycle and everything, so we can figure out where its Achilles heel is and how best to control it. Identifying the Fusarium and other fungi that are associated with these beetles. Pesticide and fungicide trials, biocontrol and other non-chemical options of control, and trap and lure development. And as I go through this, I want to point out something that it's sort of been a little bit of a criticism, and you'll see that when Zvi and Stanley speak, they're going to have a lot more data specific to avocados. They've been dealing with this problem for a number of years in avocados. Here in California, for the first 2.5 years of the infestation, we were faced with it in non-avocado hosts. It's only been since late last year that we've been dealing with it in avocados. So compared to the research that they've been able to do, our research up until late last year, August, September timeframe, was focused on ornamental trees and developing knowledge of the beetle in that environment. Now that it's shifted to avocados in San Diego County, a lot of our research has shifted to San Diego County and focusing on the situation specific to avocados. But a lot of our preliminary data that we've developed over the last couple of years is not specific to avocados because we didn't have an active infestation in avocados to learn from. So beetle identification, as Ken mentioned, it was originally identified as the T-shot hole borer. Richard Stouthammer at UC Riverside did some genetic analysis on the beetle. And when he compared the genetics of the beetle here in California, LA, Orange County originally, to the beetle, the T-shot hole borer, he found that there was a 14% difference in the DNA of those 2 beetles. Usually to identify a new species, they consider 3% difference as that cutoff for a new species. So this beetle is very, very different from the T-shot hole borer, and they're working to officially identify it and name it as a new species of Ulacea. It originated from Southeast Asia, likely Vietnam, maybe very southern China where it meets up with Vietnam. In the spring of 2014, a new population of the beetle was found in El Cajon. And again, The genetics were performed on that beetle. We were concerned because of the distance from the LA Orange County population that material had been moved, that moved the beetle down there. When they did the genetics, they found that, yet again, this is not the same beetle as LA and Orange County. It's an extremely closely related species, but it is a different species of beetle, and this one originated from Taiwan. Fortunately, these beetles are closely related enough that there's probably going to be a lot of carryover, if not 100% carryover, from what we learned from the LA Orange County population to the San Diego population and the situation in avocados. So we have a lot of confidence that our previous time spent with the LA Orange County population of the beetle was not wasted. We can learn a lot from that and transfer that knowledge to San Diego and the beetle that's attacking avocados there. So this is an ambrosia beetle. That means it's basically a fungus farming beetle. It's self-sufficient. The fungus is its food source. It does not actually eat the tree. It just bores the hole, shovels out the sawdust behind it, and inoculates the gallery with the fungus, and then feeds on the fungus. So it's self-sufficient. One female can start a whole new cycle with this beetle. The sex ratio of the beetles are heavily skewed to females, so there's very few males to females. The males are flightless. They don't leave the galleries. It's all about the females in this situation. The females emerge from the galleries already mated and then start the cycle all over again. Newly emerged beetles, they have one of two options. They either reinfest the tree in which they were raised on, or they can leave that tree and go find a new host tree to start the cycle over again. What triggers them to either stay on the current host tree or find a new host, we're not sure of. It probably involves several factors. Probably involves what is the density of the beetle on that current existing host tree. What's the status of that tree? Is it still a healthy enough tree to be a host? And there's probably some climatic factors as well that trigger when it disperses. Based on what we know from the T-shot hole borer, these beetles generally fly only at temperatures above about 68 degrees Fahrenheit. They can fly for up to an hour, about 1.3 to 2 feet per second. If you factor in very favorable tailwind situations, they may be able to fly up to 8 miles in one generation. Very, very likely that they rarely ever go that far. They probably go as far as they need to find another suitable host and then start the process over. But worst case scenario, one generation could go 8 miles, and there could be several generations in a year. So the fungi. So it's important to note that there's different fungi between the LA Orange County population of this beetle and what we have in San Diego County in avocados. In the LA population, there's 3 fungi associated with this. One is Fusarium uelacea. There's a graphium species and an acromonium species. Both of those Keith Eskelin is working on describing and naming officially. The San Diego population only carries 2 fungi with it. It carries another Fusarium species, not the same Fusarium as the LA population of the beetle, and it carries another graphium species, again, not the same as the LA population of the beetle, although closely related. These pictures just show you the 2 different beetles. Again, morphologically, visually, you cannot distinguish the 2. Only through genetic analysis can you distinguish them, and then the fungi that are associated with them. So pesticide trials, and a lot of this work was borrowed very heavily from what was done in Florida with the redbay ambrosia beetle and laurel wilt disease. Based on information that Bayer Crop Science and their researchers have shared with us, it's unlikely that neonicotinoids, imidacloprid, et cetera, are going to be very effective against this beetle, although you'll hear from Zvi that they've had some success with imidacloprid in Israel. Most of the pesticides that we currently have registered on avocados are probably not going to be effective against this beetle. And the IR4 process to get a new pesticide registered is a lengthy process, 4 to 5 years, to take a new product and put it into that process and get it through the EPA and get it into your hands. So what we've been doing, we've been looking at trunk injection and infusion trials at Pine Tree Ranch with different materials, both pesticides and fungicides, as well as combinations of the 2. Trunk sprays of bifenthrin. Let me step back on the trunk injections and infusions at Pine Tree Ranch. That is to develop sufficient efficacy data to apply for Section 18 emergency exemptions. The materials we've been looking at specifically, bifenthrin, Hero, and we've been using that because it's already in the IR4 program for other purposes. And so there's enough data, it's far enough along in the process that we're optimistic if we can develop the efficacy data to show that it's effective in avocados against this beetle, we could potentially have an application in and have that Section 18 in place for you to utilize that material by later this year or early 2016. Again, as Ken mentioned, it would require up to 5 applications a season. It's not a systemic material, it is a contact material, as closely spaced as every 15 days. And again, the bioassays are underway to develop that efficacy data to make that Section 18 application. The fungicide trials, Keith Eskelin's been doing all this work for us. It's been a time-consuming and laborious process. He started out with laboratory assays, as all plant pathologists do, using Petri dishes and growing the fungi and looking at what chemicals are effective against it, moving on to greenhouse studies with young avocado trees. And now that we are in a situation where we have infested trees in the field, those trials have moved on to field trials. So he started with more than 15 fungicides in those initial lab trials, narrowed that down to 5 when he went to greenhouse trials, and now we're looking at 3 materials in the field that show the most promise and have the best potential to be registered with the fewest hurdles to get into your hands. Tilt, as Ken mentioned, is the one that we're really focused on right now. There's a lot of efficacy data on it from Florida. Florida was successful in getting a Section 18 for the use of tilt against the redbay ambrosia beetle and laurel wilt. So we're very optimistic that we can kind of ride their coattails and get a Section 18 here in California for that material as well. It's applied as an infusion or an injection at the base of the trunk. It's laborious. It takes a couple of months for the material to move through the tree and become systemic. The good news is once it's in the tree, it looks to have efficacy for 18 months to 2 years. So you wouldn't have to do it very often, but it is a preventative, it's not a curative for a tree that's already infested. So this would be something that if you're in an area where there's a known infestation, you would want to protect those trees that are close to the infestation that are potentially going to be attacked. Biological control, so Richard Stouthammer and Keith Eskelin have made 2 trips to Vietnam. They made one in April of 2014, and they just went again in April of this year. Again, the native range for the LA Orange County population of the beetle. They have established a cooperative research program with some researchers in Vietnam, and they've been successful in securing USDA funding to help fund those researchers in Vietnam. Still trying to get through all the red tape to actually get the money to the Vietnamese researchers so they can ramp up their program there, but that's underway. On their trips to Vietnam, they've collected over 450 samples of the beetle and found associated with it predaceous thrips, several species of mites, as well as other potentially predaceous beetles and fly larvae. And so those are all being identified, and they're in the hands of the Vietnamese researchers to rear the beetle, rear these potential biocontrol agents, and see if they have any efficacy and have potential to be brought here to the US. They were just in Taiwan as well on this trip this year. The situation in Taiwan is a little different. Again, Taiwan being the native range for the San Diego population of the beetle. They found very, very low populations of the beetle in Taiwan when they looked for it in avocado groves, suggesting that there's some potential good biocontrol agents there on the island. They found a researcher who did a postdoc at UC Riverside and is now working in Taiwan. And so they're establishing some cooperative research with that person as well to continue to look for potential biocontrol agents there in Taiwan. So some very fruitful travels for them. Along with the biocontrol for the beetle. We're also looking for biocontrol for the fungus. And Keith Eskelin has been screening the fungi and bacteria that natively live on avocado trees to see if there's anything there that could potentially attack these fungi. He's identified 32 different bacteria and fungi from avocado trees. 13 of those have shown some level of inhibition of growth of the Fusarium species, most of those being Bacillus species. And this picture of the Petri dish shows you 4 different strains of these bacteria that are inhibiting the growth of the Fusarium, and one, the blue arrow there, that has no efficacy at inhibiting it. So he's going to continue to look at seeing if these can be developed into a tool that you could use to put back into your orchards to attack this fungus. The good news, if this does pan out and is a viable option, it would be an organic solution, because this is something that's endemic to avocado groves. You would just be putting it back into an avocado grove. So it would be something that organic growers could use. So it would be a very good solution. And lastly, the trap and lure technology. As Ken mentioned, we have a network of traps out. Originally, the lure that we were using was called 3250 by the manufacturer. It was very expensive, very difficult for them to produce. They could only produce it in small batches. And we had a difficult time getting enough lures to put out the traps that we needed to put out. So Richard Stouthammer did some trials with them looking at different formulations of the lure, came up with a new one, 3361, that has shown in trials to be at least as effective as the original lure, in some trials even a little bit better. So that's the lure that we now use. Richard is also looking at the trap types. We're currently using this 12 funnel trap. They're fairly expensive. Can we get by with a 4 funnel trap? Can we get by with some other trap types that are less expensive to use? And then also the trap efficiency. Many things are going to affect how effective these traps are and how effective the lure is, where you place it in the orchard, what's the temperature, what are the wind conditions, so that smell can be picked up by the beetles and they can be attracted to it. So all of those are currently being evaluated to revise this technology and potentially develop it into either trap and kill strategies, where you would actually put out enough traps to trap a large percentage of the population of the beetle and reduce the population in an infested orchard, or trap and infect strategies, where you could inoculate the traps with an entomopathogenic fungi. The beetle would go into the trap, it would pick up those fungal spores and take them back to their galleries, where it would infest the whole gallery and kill off a large portion of the population.
You were saying that we didn't have an opportunity to research it in California early on because the infestations weren't in avocados, but there was a lot of talk about how to deal with the tree once you take it down, what's the last word on that research that was done?
From what we know now, you have a couple of options. One, take the tree down and chip the material. Chipping it to about 1-inch size pieces will kill between 98 and 99% of the beetles that are in that material. It's not 100% effective, but nothing is ever going to be 100% effective. If you choose to chip it, what we're suggesting is do not remove that material from your grove. If you have the options, either cover it with plastic for several months to allow heat treatment, solarization of it, to ensure 100% kill of the beetle, or spread that material out so it dries very quickly. Once it dries quickly, the beetles will die off. They can't survive in dry wood. They need moist, live wood. So those are a couple of options. If you don't have access to a chipper or don't want it, go that route. Leave the material in larger logs, but definitely in that case, you would need to cover it with plastic for several months, solarize it, and ensure that you kill off the beetles everything.
You mentioned the traps are fairly expensive. How much does a trap cost?
Ken has been doing the ordering for us. I think the traps themselves are about $100 a piece. No? 60 a piece for the trap. The lure is about $10, and the lures last for about 6 weeks in the field. So you have to consider the cost of the trap plus the lure, and the lure has to be replaced frequently. So you're looking at 9 replacements on the lure a year, so $90 for lures plus $60 for the trap. And the traps do have a fairly limited range based on the data from the manufacturer. We think you only have about a 50-yard range around the trap where the lure is actually effective.
Tim, it appears that the spread, since we've been aware of it and its challenge to our groves, is huge. Is there any chance that, and not to minimize the importance of this, but is there any chance that that the spread is actually that fast, or are we just concentrating now and finding it? And we first found it in 2003, I believe you said. And how long, it could have been there for long. I'm not trying to minimize this threat by any means, but that's a hellacious spread. And could this be just a function, or partly a function of now we're aware of it, we're looking for it, we're finding it, and that sort of thing, even in the groves, that you can nick a tree for whatever reason, and you'll get it's obviously different, but it's the same. You'll look at that and say, okay, it got nicked or something with a picker or something like that. Is that a possibility, or are we actually having, in your judgment, a fantastic spread?
Yeah, that's a really good question. I think, I can't say from when that first beetle was found in 2003. How long before that was it here? Who knows? What happens with these pests is it takes a while for them to establish, build up a population, and then start to move. So, early on, if you recall back to those maps that Ken showed, the spread from 2012 to 2013, some of that was just a matter of recording history at that point. That infestation was already there, and it was our getting out and finding it. So that initial apparent spread quickly through LA and into Orange County, a large part of that was probably recording history at that point. The movement now that we're looking at in San Diego County is probably closer to real time, but I think there is a big factor of being, attentive and looking for it. And Doug O'Hara and I had a little bit of this conversation last night at dinner. If you think about from the manufacturer's information that the lure is only effective for a 50-yard radius around a trap, we obviously don't have traps out every 50 yards. So there's going to be gaps in our detection from the trapping. So then, when you say, well, we knew the beetle was at point A and we detected it at a trap 2 miles away, does that mean it jumped 2 miles? Maybe not. It could have been moving slowly across that 2 miles, and it just took the time to get to our trap before we detected it. And if there was no avocado groves in that area, so no one was really, if it was natural habitat and no one was really surveying that on a routine basis to look for symptoms, it'll go undetected. So, I think there's bits of both in there that affect it. And I'm going to be meeting with the researchers on Friday morning to talk about our detection trap network and is there a way that we can make that more efficient and improve it so that we can maybe not have some of these large jumps and see it more quickly? And I will turn it back over to Giovanni.
Okay, thank you, Ken and Tim, for that update on the current outbreaks in California, the differences between the one in San Diego, the one in Los Angeles, and all of the efforts that the California Avocado Commission is doing on behalf to track and combat the current outbreak here.